into become a recording. Okay, got it. Become a research professor at Caltech. So I did my undergraduate study in Taiwan, and my major is pharmacy. And when I was doing my pharmacy degree, I really enjoyed organic chemistry. So after I graduate, about 4 years later, I want to come to US to do PhD study. And because at the time during the undergraduate training, I realized, you can be a doctor to help patients, but a lot of people suffering for certain disease, like even cancer and the brain disease, the neurodegenerative disorder, we don't have good drug. So I figure, okay, maybe I can come here to study more and also to figure out how people need to come to US and become, developing interesting treatment. Because at the time on TV, I saw the cocktail anti-HIV therapy from He Dai. So I was thinking it's a really simple concept, but how can, but this concept was developed in the US. So just as simple as that. And luckily, when I applied for the medicinal chemistry degree, because I'm interested in chemistry, even though one of my medicinal chemistry professor in NTU told me, oh, you are a girl, so you should go study pharmaceutics. Don't do medicine because it's too tough for a girl. But, sometimes you should listen to advice. Sometimes you should listen to your heart to figure out what you really want to do. So I spent 5.5 years in Minnesota, Twin City, for medicine. And then I finished the PhD study, and I enjoyed research, and I want to learn more, because when we are reading the paper, later if you do research, you will know some of the paper, there's this hierarchy of, okay, which journal is better, all of this. And to discover a new function of a protein or a gene that people don't know, you actually need to use more in vivo or so-called model organism. So after I finish my medical training, I know how to do tissue culture and how to do, to develop drug in test tube, but I don't know how to move to the next step. So I choose Caltech biology department as my so-called postdoctoral training. So after you finish PhD, there's another 5 years of training period. And at that 5 years, I actually learned how to do biology and how to do high throughput screening and how to use proteomics as a method to discover drug or do drug discovery and find biomarker. Then luckily, I got my first assistant professor job in 2012 in Harvard UCLA Alabama in Torrance. In 2019, I got the chance to move back to Caltech as a research professor, and also now in charge of proteomic facility. That is one of my dream. In the way, I think it's very important to be able to find every protein's function. We still don't know a lot of protein's function, believe or not, even though the research has been carried out for so many years. Okay, so now let's go to the, before I start talking about the ERT project for MPS 3D, so that's the short for mucopolysaccharidosis. This is an interesting project that even me, as when I finished my postdoc training, I learned, you can find a protein as a drug treatment. And these are the people actually make this happen. So Phoenix Next is a company which started by a mom. The mom has a kid which has a disease, is MPS 3C. And Alabama is where I was when I started my first position. And in Alabama, actually, there is a very interesting story. Like the MPS, I haven't explained the detail, but there are different subforms, like 1, 2, 3, 4, 5. The one I'm going to talk about is 3. But before, many years ago, 20, 30 years ago, there's zero treatment for those horrible neurodegenerative disease for the kids. And the kids, when they are born, they don't have the functional protein or this so-called enzyme. And Alabama actually was the first place to discover MPS 1 treatment. Also, it's an ERT. And the reason was the father of the kids called Ryan, and now they have a foundation called Ryan Foundation, realized there's no treatment for his son. So what did he do? He go collect money from people. And he go, so he collect basically donation from people and find a doctor and a PhD who was trying to study this pathway. So his son is still alive because of him. Yeah. And when we start the project at Biomed, oh, since we can do this for MPS 1, right? How about many other subforms of this disease? 3D is one of the rare one, but again, there's zero treatment. So we can apply similar idea, just purify this protein as a drug. And those are the people, and Washu is Patty is the one doing animal study, and Caltech now is my current lab. And the people I highlight in blue was like move with me in Caltech and continue this research. And Feng already went back to China. He started his own lab as an associate professor now. And San is working in a pharmaceutical company in Beijing. And Stephen is in Patty's lab now in Washu. So, and Sri and Gio is the, in charge of the company now. Gio is the mother I mentioned who started the company. And why it's important, because they are the one actually got the money, got the funding, the support from NIH to help develop the treatment. And those are the other people like the Dr. Emily and Gio Morris. Those are the so-called grant manager or program manager for under NIH, one of the institute called Institute of Neurological Disorder. So their job is to give money to help give money to the right people to do the right research to find a treatment for neurological disorder. And we have meeting with them like every month to make the progress. So I really feel this is a big teamwork, not just one small lab. See, it's so many moving parts to make this work. Okay, so what is MPS 3D? So this disease is caused by absence of an enzyme called glucosamine N-acetyl 6 sulfatase. So for short, we call GNS. And because the kids was born lacking functional enzyme in their body, and an enzyme basically is a protein inside our body and will carry out a reaction. I will show you the reaction later. So without this functional enzyme in the body, we will have this accumulation of substrate called heparin sulfate. So you can see in this cartoon, just want to show you, normally this complex substrate, when in the healthy person, those will get degraded. Can you see my mouse moving? Oh, good. And then in the disease or in the patient's body, they don't have the functional enzyme. So you can see we call lysosomal enzyme deficiency, because those complicated substrates need to go inside our body into a specific organelle called lysosome. So I don't know if you heard about mitochondria, you have the nucleus, but there's another special organelle called lysosome. And this organelle is important to degrade some protein or protein complex, because inside this lysosome, there are a lot of proteases which chop out the protein. So when the kids don't have, and GNS is one of these enzymes inside lysosome. So when the kids was born lacking GNS, you can see there are a lot of substrate being stored in the lysosome. And these actually make lysosome dysfunctional. And that's how the neurological defect comes. And for the MPS 3D, it's focused on neurological, but MPS 1 or 2, those actually has both, has systematic defect and also the brain defect. So for 3D, the difficult part, since it's only in the brain, the defect, so the treatment we develop has to be able to go to the brain, right? Because the MPS 1 in the beginning, when they develop the ERT, they only need to inject IV, then they can treat other systematic symptoms. But if the enzyme is not sufficient to go to the brain, then we cannot treat the 3D. So, okay, so I just explained accumulation of these substrates called GAKE in the lysosome. So this is not to scare you, but just to show you like how complicated and what is the amazing chemistry in our body has to happen to keep everybody healthy. So you can see those like six ring circle. Basically, these look similar to like glucose. So when you take the sugar, take any sweet thing, you have glucose. Glucose looks similar to this. So inside our body, there are these multiple sugar-like or molecule, they connect to each other. and they have specific function. They exist for the reason, but during the process, they have to be degraded to be recycled to make our body functional. So among the way to recycle or to get rid of those polymer, like the chain, right? They connect each other. You can see like A, B, C, D. So this different subtype basically is named after the certain enzyme or protein that is required to do that specific chemistry. And I think ABCD just the time people discover the disease. So like 3A is the first one got discovered and they call 3A. And then later they found, oh, there's another type of mutation we'll call 3B. And the mutation basically is when you know, the protein is made, there's this DNA sequence. So the mutation majority is in the DNA sequence, genetic disease. And the DNA sequence change cause the protein sequence to change. And this sequence change in the position that actually is important for this particular protein or enzyme's function. Therefore, this change is not good because, you know, it make the protein not functional. And 3D is the one, basically, you can see this is the particular, the substrate, this particular enzyme need to be able to cleave. So we'll release this OSO2, so it's called sulfatase, get rid of the sulfate group. And then when we try to do research in the lab, one very important thing you need to develop is called assay. What is assay? Like, for example, there is assay to determine protein concentration. That's called Professor assay. And Professor is a guy's name, a scientist who discovered, oh, you can use that dye and the dye bind to the protein as a way to read out protein concentration. And since our enzyme, MPS 3D, the enzyme GNS, is required to remove the sulfate. So we try to find what are the similar idea. Okay, if I can know, I put this enzyme or this solution with certain thing, I know I have a active, a functional, working enzyme. So we need to develop assay. And I don't need to explain too much. I just want to know. I tell you, in the lab, when we say assay, or there's basically a way to detect something, right? And assay can be detected by color, can be detected by like you can run things and detect by signal on the the reader, which or HPLC, like something you can have a signal to detect, or you can simply detect by measuring the amount, but there you need to have certain things so you know whatever you are detecting. So this is the assay. So this, okay, so the previous assay, the one I just mentioned, these are called absorbance. Since I put it there, just explain a little better. So this is reading at the light. There's it tend to yellow color. So when you don't have functional GNS, the solution will be clear. But once you put the functional enzyme, the solution will become yellow. So you can, based on the intensity or how intense the yellow, you can have light yellow and very dark yellow, tells you how much the enzyme is functional. So this is so-called absorbance. But this assay is not as sensitive. So the next assay I'm going to tell you is complicated. Can you see so many steps? And the reason we also use this particular assay is because the substrate, which is the things on this side, I'm circling this is more similar to what is the gate or substrate in our body. This enzyme is supposed to get rid of. So in this case, this is a beta substrate. But the problem, can you see, is more complicated because you need so many steps. But sometimes when you design the experiment or the assay, there are pros and cons, right? So you want to figure out, you want to think, okay, I have 3 different choices. Why did I choose this one? And the reason in the end we use this one main reason is this is more similar to what's happening in our body. So when I explain the disease actually is caused by the kids is born or lacking this functional enzyme, right? And the treatment, there are different treatments. For example, there's a gene therapy. So I don't know if you guys heard about it. So you can deliver the functional gene, that's called gene therapy. But sometimes gene therapy doesn't work for all of the disease, right? So there's another way to treat. It's called enzyme replacement therapy. You can call basically the protein therapy compared to gene. You basically deliver a functional protein. And in a way, it's a little bit harder because for gene therapy, you only need to know the sequence of that gene you need to deliver. For protein, you actually need to isolate it. You need to purify it. And there are different ways to purify protein. And because this protein GNS is a protein that will undergo glycosylation, like glycosylation basically is a sugar molecule put on the protein. So this protein we cannot purify from E. coli. So we have to use another system called Chinese hamster ovary cell. So that's CHO cell. So that cell is being used in pharmaceutical company. It's a platform, it's a cell that you can express the gene in that cell, and you need to manipulate, you need to try different ways. You use that cell to produce the protein you want to purify. And so the picture shows here, like there's a blue circle, so G418, there's a red circle called MPS, methotrexate. These are like two different type of systems, also called plasmid. And you can deliver to this so-called CHO cell I try to explain. Basically, it's like a platform or a media that you use to make the purpose you want. And the approach has pros and cons. The G418 is quick, fast, but the yield is low. MPS is a way to force the cell to express a lot more protein, but it takes a long time, like it takes 3 months. The G418 only takes 2 weeks. You can already see if it works or not, but MPS takes longer. And in this paper, we already published last year, we actually compare both because we do G418 to make sure whatever we are thinking will work. Then once it works, then we go to do the longer one. We do the MPS, can purify more protein. And once you put those so-called plasmid in the cell and you express the protein, then you need to find a way to purify. So this purification, and you can see we just put a simple column here, but this actually is a more difficult step because different protein has different so-called stability, like it can tolerate different conditions. So you kind of need to play around with many different conditions. And with this simple column, you can put different resin inside the column to help you to purify. And purify, you think about what does purification mean? Purification basically is you express your protein in this soup of the protein, right? And your protein is only one of them, but there are like at least another 10,000 other protein exist in the soup. And we already use the plasmid to call overexpress, meaning you try to force your cell to make more of the protein you want, but still it's within a soup, right? So you need to have a way to basically get rid of the impurity, so other protein you don't want. So that's purified. And once we purified it, we basically put into patients. So we have MPS 3D patients cell to see if the protein can correct the defect in the cell. And we also put in the MP3D mouse. So mouse model is very important for drug discovery, but unfortunately, you can think mouse brain and human brain, there's a huge difference even in size, right? So there are many things the mouse cannot recapitulate the brain disease. And that's actually one of the difficulty why a lot of drugs that for brain disease failed, because we use the mouse model to do so-called preclinical study, but then in the end, it only works in mouse and doesn't work in human. Or we actually don't have good mouse model for a lot of neurodegenerative disease, just because what I just said, yeah, no, the brain is different. So we don't recapitulate what happened in human. And then in use this, we actually give the purified protein to patient cell and to the MPS 3D mouse. The mouse does not have functional GNS. And then we do the activity, the functional assay. So basically you can see the first is GNS carrier. So this is so-called wild type. And the next one is the mice, the disease mice, and treat with so-called vehicle. VHE is the vehicle. So if you compare the wild type and the 3D mice, you can see the disease cause accumulation of that substrate, right? It's 100 versus 50. And then when we treat with the enzyme, this is a smaller dose. After 7 days, you can see the reduction of this accumulated substrate. So the whole paper is trying to describe the whole process from the beginning to the end, like proof of concept, this work. And then since that work beautifully, oh, and I forgot to mention, so in the paper, we only use so-called newborn mice. So the baby mice, basically, because then the mouse weight is smaller, so we don't need to put so much enzyme. And we use that as a proof of principle. Then once we see that works, and then we purify more protein, then, and this mouse study is done at a petty distance lab at WashU, and we, phone and sign, we purify protein, and we send in the protein. And this figure, I just want to show you how the treatment basically directly inject the purified protein in the injection site on this figure. So this is the mouse brain, and we inject in the section 2. And after the treatment, then they cut the mouse brain into 3 sections. Oh, you inject when the mouse is alive, so it's a surgery. So this is not something easy to do, because, but they have the expertise and skill. So the live mice, then they need to have a surgeon to implant a cannon, and then they can inject our protein. And then, of course, then let the mice move around. And if you need to repeat the dose, you repeat. And if not, we just after a few days or a few months, depends on the study design. Then you collect the, you need to sacrifice the mice, unfortunately. And then you collect the mouse brain and you cut and to do the assay to know if the protein actually distribute to the mouse brain. So the conclusion here we want to say is the enzyme, the GNS we purify, can distribute to 3 sections of the brain, even though we just inject in this section 2, these dots here. And we cut into 3 sections and we do the enzyme assay. So this is different from the heparin sulfate I mentioned. This is basically using that crazy, like multiple step assay to determine the enzyme activity, GNS activity in the mouse brain. So we are happy to see, and we can see the dose dependent effect. So the first dose is only 3 microgram, and we increase to 30, so 10-fold of 3 microgram, and 200, not 300, sorry. So we can see the enzyme activity increase in a dose dependent manner. And then the other important part, which is the gag, I show you for the the newborn mice study. We can see we can also reduce this bad gag in the treated sample. So this actually is quite successful to see the effect of the enzyme. And then the assay or the gag reduction assay I just show you, it originally is sent to University of San Diego. They have a mass spec facility who is dedicating measuring this gag. But since at Caltech, we also have the mass spec facility. So we decide to optimize this assay. So what we can do now, actually, we can increase the sensitivity by 1,000-fold. And what does that mean? It means the same amount of the gag, you can dilute. You can take 1 and put with 1,000 of the whatever buffer, you can still detect it. And the reason we want to do this is we also want to detect the CSF, the mouse, the fluid in the brain, and these are tiny. So the more sensitive the assay, the more accurate the assay can be. So we are happy we can do this assay now at Caltech. So I'm going to try to explain what proteomic analysis is. But I think this figure is a little bit too complicated. I will go here. So basically, maybe I go to I can come back. Here. So the proteomics, which is trying to detect all of the protein in our body as much as possible. And there are 2 types of proteomics. Like one type is called bottom up. That means inside our body we have so many protein, right? And bottom up means we need to chop our protein into peptides, and we assemble the information from peptide. Peptide is small, chopped protein, and to know, in this tube, how many proteins are there and in what amount. And the other approach is called, and then this need to be analyzed by a machine called mass spectrometer. And the other approach is called top-down, meaning we don't chop the protein. We try to analyze the protein when it's intact. And the method number, the top, the A method, it's a little bit easier because the way the method works, you only need to analyze a small piece of the peptide and to know your protein amount. The top-down is harder, but there are a lot of people trying to, there are some group of people trying to make the top-down improve as much as we can to detect as many proteins. So for a bottom-up, now easily, if you have a tube of the human say sample or lysae, like in serum or in the brain lysae, we can easily detect like 6,000-7,000 proteins from bottom-up. From top-down, now I think we only can detect 1,000 to 2,000. So there are still a big difference. Okay, now go back to what proteomics and why do we want to do proteomics? So what we did in this case is we want to detect all of the protein in the diseased mice compared to so-called normal mice. And the reason is we want to know, in addition to the accumulation of this gate, what are the difference between protein expression difference between the wild type and the diseased mice. And we can find something called biomarker, because if we see things is different, and then if we treat, we give the mice, or no, in the end it will be human, right? The treatment, if the difference can go back to normal or so-called wild type, which is normal, then that is a good way to show the treatment work. So if I look at more protein, then I have more confidence to know more things got changed back to normal. So this is basically so-called heat map. And red meaning things increase in the disease, decrease in the disease, because it's a wild type versus 3D. And it's green, it's increased because GNS is the gene, right? So if you knock down, you should see GNS decrease. And then when we treat the diseased mice with the protein, we see the replacement. So we see the GNS level go back to the normal. And similar thing for other protein marker we can detect. And you can see those are other so-called lysosomal marker, those like the one we make it bold. Those are the protein people already know in different type of disease will change in the lysosome. So we are happy to see so-called similar result to others. But many other protein that other people has not published or has not told the community, oh, it's important, we see. So then once you do this mass-based experiment, normally what you do is you go to verify, see if this CDC68 is this important. And then if it's important, then you can use this as a good biomarker. And then the next thing you will do is you will say, oh, I have 100 protein change. Then you can do a thing called pathway analysis to know, does this 100 protein function or work in a similar way? So when we do this, you can see, we see those proteins are important regulating cell death and regulating some neuronal function. So that's also, and then those, it's a way to kind of help you to figure out what to do next. Yeah. And now the ongoing thing happen in the lab is, or with the company collaboration. The first one is the assay I just told you, we want to be able to do the CSF. So that is the current thing. We already did the mouse brain, but we want to measure this spinal fluid in the, from the mice. Then we know the purified protein work in mice, right? But to put the protein in human, we cannot prepare in the lab. So now we have a company is making the clinical grade recombinant GNS protein. So once this is met, then we need to, of course, make sure it's safe and also has efficacy. Then we can start the first human clinical trial. So actually for this project, I went to FDA before COVID, like 2019 or 2020. I don't remember exactly. I think 2019, no, 20 is already COVID. Right? So 2019, I went to FDA and with Gio and Sri, with company and Patty, company people, to discuss with FDA, how do we do this clinical trial? Because one thing I forgot to mention, the MPS 3D is an ultra rare disease. Like so far in the US, we only know there are 5 kids with this disease, right? Can you think about how do you do clinical trial? You cannot say, oh, I split this into 2, right? And 2, you get a treatment, 2, you don't. That's not ethical. So, and luckily FDA is quite supportive. So they understand these are like not like cancer. So you can have a lot of patients, or like COVID, you have a lot of patients, we can do the clinical trial easily, right? So, and so no, we already come out with the plan with the FDA, but no, the GMP grade, currently we already prepared and we are doing the safety study. So as soon as safety study is done, then we can start to recruit for the phase 1 clinical trial. What else? Okay, for the mass spec result, I just show you, we need to verify by different methods. And we are also doing another project. Is basically, I try to explain mouse brain is not human brain, right? There's another way you can create human mini brain from the induced pluripotent stem cell. So we already have the patient's fibroblasts, and we already make them into the iPSC, so induced pluripotent stem cell. And we are in the process to make the mini brain from the stem cell. And the same thing, we can use this to also monitor whether or not the treatment is good. And the important thing is also, can we find other things that we don't know when we use the mouse model to do the study? I think that's it. And I already mentioned to you who are the people. And of course, I have other people in the lab, but I didn't mention those who are not working on this project. But I'm happy to answer any question or any other question you might have related to this or related to research in general.
Okay, thank you so much, Professor Chou. So everyone, if you have any questions right now, you can either type it in the chat, or if you want to, you can unmute yourself. Sure. Yes. Oh, Professor Chou, like I can read out the questions if someone type in the chat. Yeah.
Okay, good.
Mm-hmm. So would anyone want to like ask anything about like either this project or about like anything general about research or about science?
You can raise your hand.
Yeah, or you can raise your hand. Yes.
I have a question. So
yes,
how long does it take to go from like phase 1 research to getting the approval from the FDA?
Okay, that's in general, if you go to Medicaid program, they normally tell you it's 10 years. But there are, so that's a general concept, because after phase 1, and phase 1 normally is to look at for safety, right? And there's a phase 2, and there's a phase 3. So that's why the COVID vaccine actually is amazing. Like the way we can do this in this country, this phase approval, including everything, the safety, right? But for normal drug, it's 10 years. And for brain drug, in other type of brain drug, it's even harder, because like those clinical trial is longer. Can you imagine, how do I know if so-called Alzheimer, how do I know the treatment? I don't know if you hear the story, like the FDA approved a drug and then they don't know if it's actually functional. Those are all sad stories, because when they spend a lot of money to develop, they must see some effect, otherwise they are not going to put. But when you try to do the trial, who are the patient or who are the people participate also matters, right? So for brain disease might be even longer. But for others, because we have a small cohort patient, so might be a little bit faster, because we can negotiate with FDA but you need to make the plan first. They need to approve whatever you plan, it makes sense. No. Yeah. Because you can think if I tell you 10 years and the kids, now the 5 patients, some are 9 years old and 2 twin sister is already 15. So by the time approved, they are already there. If we do 10 years, already 25. Yeah.
Thank you.
No problem.
Professor Chou, Nadia asked in the chat, like, what are the symptoms for this MPS disease? Like, what are some general symptoms?
So for MPS 3, in general, it's a brain disease. So like and it's progressive. So they actually in the beginning are fine, and at certain age you start to see like basically they cannot function properly, and some of them in the end may not be able to walk properly. And MPS 3D is mild. Like I think the 2 twin sisters we know, if you look at and talk to them, they actually seems to know normal when they are in age 16. But there are certain things they start to not be able to do. And for MPS 1, for example, that one is very severe. So when the kids are born, like some of them, you can see the brain is already bigger, and some of them cannot already move when they are a baby. So there's a big spectrum. Yeah. And MPS, you can think in a way, it's basically Alzheimer in or for MPS 3 in general, because it's only focused on brain. For other subtypes, it's more severe. But MPS 3 is more like Alzheimer in kids. So because they are focused on the brain, so they actually can do many other things. They can play basketball, they can do all of these things other MPS subtype cannot do.
Would anyone else would like to ask any questions in general about anything?
How about you guys tell me what? Okay, go ahead.
I just have, it's not really about research, it's about Caltech. So as a Caltech professor, what does Caltech do that's different, that makes it so pristine?
Oh, I have no idea. Well, actually, how to put this way? Well, just I feel so. I was a postdoc at Caltech 2007 to 2012, right? So when I first arrived Caltech at the time, I was actually quite shocked as how easy collaboration between the lab can happen. When I was in Minnesota, collaboration doesn't seem to be that easy. Like people might not be as open-minded to say, oh, I want to work with you to develop something together. So there is one thing I feel the environment encourage people to work as a team, not even in the same lab, in different lab, right? And but we are small. So the downside is Caltech is smaller. So for research part, certain things you kind of are the only person you are working on. But you can think about, like when I try to explain the essay, you can have things that you don't have. You can work with other lab to develop, right? And some people might come to my lab, oh, we never done this. Can you help us do this? So most of the time we'll say yes, unless we also don't know how to do. So we can help together. So this collaborative environment, I think it's one of the key things. And in general, I feel like even the big professors, people are willing to talk to you, and in a way, like in another bigger place, people are more how to call that way, like hierarchy, right? And so for that, when I interview graduate student, like I haven't interviewed undergraduate, I interview graduate student, I always tell them, Caltech is unique, but you need to be self-drive. So you are the one decide what you want to do. So you don't want to sit there, wait for your advisor or your professor to tell you, hey, you need to do this. So that also encourage new discovery. Like for graduate student, you come in, regardless which division you apply to, you have the freedom to choose any lab you want to, any PI you want to work with. That also is relatively unheard of, because in other university, when you come in, because it's this division paying you, right? And they want you to only choose the professor in this department or division. So that this uniqueness is also in my point of view, is it helps people to be wanting to do things they think is important, and you are the one doing experiment, and you will see you can add like in the summer doing experiment actually is quite tedious, and it's a lot of time commitment. And how do you do it better? One is you need to be willing to commit for the time. A second is you love it. So if you love something, then you will spend time. Or you can go watch a movie, or you can go read a paper. There's a different choice for the time you decide to do. And for example, there's a project at Caltech started by 3 graduate students and in 3 different labs. And when they start the project, they actually didn't ask their advisor yet. They just, oh, we have this jellyfish and we want to see if they sleep. And this actually was on the news 2018. Like they discovered jellyfish can sleep. But the reason they got interviewed by so many press is not because of that. It's also because they actually initiated the project themselves. It's not the advisor say, oh, this is interesting, we should work on this. Does that make sense? So I think that's my point of view, but you can also, if you're interested, I can connect with people in my lab. You can ask them what they think,
Okay, thank you.
No problem.
That's the most interesting question today, huh?
Well, that's the one important. I'm kidding.
And Professor Chou, I have a question like myself. Because you said like MPS 3D, like it has such less of like the like such a small community of like the patients. Like why did you specifically choose to reach out to the not reach out, like why you specifically choose to pursue your research on this specific disease?
Okay, good question. So the long story, I will not tell you. Short story. When I was in Alabama in Torrance, so Patty Dixon is the person who actually kind of trying to develop therapy for MPS. And 3D at the time, they need somebody to help them. See, like I talked about collaboration to purify the protein. So I just basically helped them interview postdoc, but unfortunately, no, they didn't recruit the postdoc who we think is good for the project. So then they asked me for help. So can you help us to do this? I said, sure, I love purified protein. I didn't know purified protein as a drug before I came to Harvard UCLA. So we are happy to help them. And at the same time, it's also when we look at it, I think one of the main reasons we only know like 5 or 6 kids now in the US, it's because it's really hard to diagnose. You say somebody asked me about the symptom, right? Because those kids in the beginning, and when I went to the MPS meeting, you will hear a lot of parents will tell you those horrible stories. They go tell their doctor, oh, I think something wrong with my kids. And the doctor was, oh, this is just growth pain, or this is just something. They ignore because these are rare. So if the doctor does not have the training to know these are one type of disease, they basically wait until later being diagnosed, or some of the people might not be even being diagnosed until very late, right? So, but the good thing is now we can do exome sequencing easily. So, so I do think we will find more patients. That's one thing to say. But the other thing is, if the disease has a lot of patients, the big pharma is willing to work on it. So then you don't need academic to basically compete with the big pharma. So for rare disease, NIH also has an emphasis, because big pharmaceutical company is not going to put a lot of money to develop, because for them, there's no profit. I mean, they don't make money, right? But for research for NIH, they know those patients also need help. So that's why I do think it's still important. Like the disease relevant for P97 that IBM PFD, it's also a rare disease. It's not as rare as MPS 3D, but that's also a rare disease. And I do think those are what NIH can help, the National Institute of Health. They can help to provide funding. And for us, the researcher, is if whatever we can do can help the small company to be successful to develop treatment. I think that's what the uniqueness. And the big company knows how to do things that most of like already be developed, right? So for the difficult things, it's the lab or so-called academic lab should be the one willing to spend time and get a resource to make it happen. Does that make sense?
Yes, yes, it does. Okay. I think Amelia, you have a question before Jason, but I think we didn't answer your question yet.
No, I don't have
it's okay.
questions.
Okay, yeah, okay, thank you. Do anyone else have any questions in general about like you want to ask? You can like type in the chat or you can just speak it out.
Or raise your hand if you already
Yeah.
eat mooncake. No, I'm kidding. Or tell me what's your favorite mooncake?
Oh yeah, would
Forever.
anyone want to share what he did today with Moon Festival?
I don't know, I'm kidding. It's okay.
Okay.
Yeah.
Okay.
So in your foundation, let me ask you. So then you guys meet, how often did you guys meet?
Like so because we only started this program like this summer, so
Okay.
this is like our second meeting and we're still planning to have it as monthly. So like my eventual goal is to reach out to the actual patient community. So I've been trying to contact some patient associations and see if we can connect with one of their families, like patient families, and then have them share the actual like their experience with the disease, and then we can share what our past how we have like how our past work of us trying to raise awareness for like certain rare diseases. And then yes, so it's to like a feedback to the community, and then to hope like more people understand how like understand the importance of like this like to like the importance of knowing these diseases. And yeah, and okay, so last call of like if anyone wants to ask any questions.
Wait, last thing, would you mind like private messaging like your email or something?
Oh yeah, sure. Oh, you don't have my email? Let's see.
Oh, Tracy, I can share with you if you like. So
wait a second. Yeah, okay, I put in the chat.
Oh yeah, so if you guys have any questions in general, you can also reach out to Professor Chou. Okay, so if you guys don't have any questions for now, then we'll be proceeding into the group project part. So Professor Chou, if you could stay, then our event will go until 5 p.m. and around 4:30 we'll be starting like a group presentation. So each of us will be presenting like a short slides about like their thoughts and reflection on today's topic. But if you are too busy, if you have something else, it's okay, and I can send you like a recording of our
oh yeah, you can send me. I didn't know it's a 2 hour, so I
it's okay.
Get Peter.
Get Peter. Okay.
Yeah, but
it's okay,
everybody, feel free to reach out. The goal is I do feel it's important to let people know everybody can help, right? And if you're interested in the research, feel free to reach out. Okay. Take care.
Yes. Okay. Thank you so much. Yeah. Okay. Have a good day.
Bye bye.
Wait.
Thank you.
Thank you. Bye bye.
Thank you. Have a good Moon Festival.
You too. Bye bye.
节快乐。
也中秋节快乐。拜拜。
Okay, so now we'll be proceeding into the group project part. So after I introduce you guys to what exactly is the group project, then you guys will be entering each breakout room based on the group numbers you have. So when you first enter into the breakout room, you'll be going over like a short icebreaker activity. So you will first introduce yourself, what's your name, where you're from, some hobbies of yours, and you'll share one fact you know about neurological disorder. So like maybe some experience or some personal experience with neurological disorder, or like something about neurological disorder. So you have to share one fact about it. And then home scavenger hunt. each of the group leaders will be sharing you guys like the instructions for this activity when you enter into the breakout room. So about the group projects, I will be going over like what each slide is about, and then each of you guys will be assigned like each slide and then by your group leader. So you'll know what slide we'll be working on. And so for the first slide, we'll have a person to design a cover with your unique group name. So you come up with your group name and then your group member and a design that matches today's topic. So today's neurological disorders. And the second slide, you will have list out a few key points of today's speech. So what you heard from Professor Chou, and you explain briefly explain what is the neurological disorders and then maybe some of its symptoms or causes. And the third slide, and someone else in your group. So we encourage you to have like two people to work on the third slide. So one will be searching out more facts about the neurological disorders, and then they can list out the facts on the slide. And the other person will be explaining the significance of understanding this disease. Like why should we even trying to understand, like what's the point of understanding this disease? So lastly, you have a fifth person to work on the fourth slide to explain the method of treatment that was introduced by today's speakers. So like what the enzyme replacement therapy that Professor Chou talked about, like you don't have to explain it in detail, but if you could briefly like summarize what she just talked about. And on the fifth slide, it's your personal reflection. So each of you person will write their own. So like you have your name and you write a brief paragraph or 3 sentences on your reflections on today's event. So what do you think about this neurological disorders and why, like your general reflections about today's event? So before you enter into the breakout rooms, you'll find each of your group slides under the Google Sheet that's been distributed out in our emails. So you'll find the group sheet, like your group project like slides under here. So you will be working on that Google slide. So you just click on it, and then the group leaders, you guys can assign each of the members what slides they can work on. And if you guys have any questions, you can call me into your breakout rooms and then I can answer it for you. So if you guys could, please enter each of the breakout rooms. Okay, so I just opened all the breakout rooms. So if you guys could like enter the breakout rooms based on your group number. So room 1 is group 1, room 2 is group 2, etc. And if you guys have any questions, then you can ask me. And before you enter, so we'll be coming back at around 4:30 or 4:25, 4:30. At that time, I'll be letting you guys know when we're about to come back, and we'll be presenting the slides to everyone. And we're going from group 1 to group 7, and you guys will present your slide.
Remember to come back.
Yeah, remember to I'll close the breakout room, so like there'll be an alert of like you should be coming back.
Okay.
Yeah, so please join your breakout rooms based on your group number.
See you later. Enjoy.
Yeah, see you later. Okay, so we'll be starting from group 1 to group 7, and you will be presenting your slides based on how you guys arrange it. So if group 1, you guys could share your screen and you guys could start your presentation.
Eric, would you mind sharing your screen?
I don't have the thing.
All right, I have to do it.
Okay, I make you a co-host. Okay. Thank you. So this is a group 1.
This is group 1, yes.
Thank you. 你们报一下名字哈。
Hey guys, we're group 1, and our team name is His Majesty the Supreme Leader Adjacent Subjects, and we'll be presenting MP3.
About MPS 3, The causes are the lack of enzymes that break down a large complex sugar molecule called the heparin sulfate, and some family history of MPS 3 can increase the risk. And some symptoms of MPS 3 are physical disabilities such as improperly walking and altered moods such as often being irritated.
So some facts about MPS 3 is that it is a very rare genetic condition that can cause very fatal damage. It can cause death, and it can be do very damage to the body. It can be also known as the Sanfilippo syndrome or the childhood dementia. It can cause by the lack of enzymes which break down and recycles heparin sulfate. And that's the accuse of the sulfate can cause damage to the cells of the CNS, and which can lead to the fatal damage. It is mostly caused by accumulation of mutations in the genes, and children with MPS 3 usually appears healthy at birth, but then later shows developmental delays with mental development peak at the age of 3 to 6.
For the treatment of MPS 3, there isn't any cure. But the most promising treatments are gene therapy and enzyme replacement therapy. Now, for enzyme replacement therapy, it helps increase the enzymes missing in the body, which can slow the progress. And for gene therapy, it does something similar to ERT.
Okay, so now we're going to go over what we thought of today's disease. So one person in our group found it very touching and awe-inspiring that the professor decided to research this disease despite being so rare. Another person thought it was very useful for the human society, and it's very shocking some of the symptoms that the patients has to endure. And it's very important that we are researching this disease because not many people are ensuring that this disease, and it is very detrimental for the health of the human body and a family. And that's about it for me.
Okay, thank you, Group 1. So would Group 2 share their slides and you can start your presentation? Okay.
Hello, so we're group 2, and here's our neurological disorder sets. So some of the key points were that MPS 3D is a neurological disease which affects the brain, and it's a recessive hereditary disease caused by the absence of the enzyme. I'm not going to try to pronounce that. Glucosamine 6 sulfatase, and affects a person's ability to break down large sugar molecules. And some of its symptoms are loss of movement, dementia, seizures, deafness, and loss of vision.
Some extra effects would include that the life expectancy of children affected by this disease would be 10 to 20 years. Major causes of death due to this disease would include heart disease and airway obstruction. A family history of genetic disease can increase the risk of developing this disease. And extra mucopolysaccharides and urine detected by clinical tests is the first step of identifying whether someone has this disease.
This type of a disease is an autosomal recessive type of inheritance. Basically, it just means that the gene is located in any chromosome besides the X and Y chromosomes. And because this disease is caused by a mutation in the gene sequence, the expression of this gene causes proteins that are made from this expression unable to be functional, so they're not able to perform actions like they were supposed to. And because of this inability to express functional proteins, the enzymes that are made from these proteins are unable to break down the certain molecules that are needed for the body.
Methods of treatment. Different methods of treatment that the speaker talked about today was gene therapy and enzyme replacement therapy. Some attempts at gene therapy for MPS were mostly in animals that involved injecting to the second part of the brain. The other types of treatment would be enzyme replacement therapy, which is used to correct the enzyme deficiency of the enzyme glucosium.
And here are our reflections. So I wrote that from this event, I learned about the neurological disease MPS 3D and its effects on the people affected by it, as well as what kind of treatments are available to people who have it. I found it really fascinating how the absence of just one type of enzyme can affect a person's body so much.
I learned that MPS is a neurological disease that affects the brain and it's caused by gene mutations. The treatment for this is therapy, and children who are affected by the disease have a life expectancy of 10 to 20 years. And some symptoms are loss of vision, loss of seizures.
I learned from the damage of how much damage the mutation from the gene sequences can cause the person, and not to mention the lingering effects that these have on the descendants due to gene inheritance, and the various types of treatments that have been used by doctors to help treat this, along with enzyme therapy and gene therapy. Enzyme therapy being them trying to help combat the deficiency of the enzyme, and gene therapy to help fix the gene sequence that has the mutation.
I learned that the neurologic disease MPS 3D is because of an enzyme deficiency of glucosamine and can be treated with gene therapy and enzyme replacement therapy. I also learned that the disease affects the brain but is hard to identify since this disease is regressive. Children are most likely to be diagnosed with this disease, and with this disease, the life expectations of children are only up to 10 to 20 years.
Wait, sorry, is anyone continuing the presentation?
So down with the group 2.
Okay, yeah, okay.
Here.
Yeah, it's okay. So can group 3, could you guys please share your screen and then we'll continue on the presentation.
Yeah.
Okay, thank you.
Can you guys see it?
Yes.
Hi, my name is Aubrey and my group members are Jaden, Darren, Alice, and Hans. Our group name is the Neurological Crusaders.
So what is MPC3? It's a genetic order that makes your body unable to break down large sugar molecules called glycosaminoglycans, or more specifically, a GAG called heparin sulfate. The causes of it is caused by missing or deficient enzyme. I'm not gonna say that, it's too long, and inherited in an autosomal recessive manner. Some symptoms of it is progressive dementia, aggressive behavior, hyperactivity, seizures, deafness, loss of vision, unsteady feet, loss of motor function, loss of language skills, and loss of some or all hearing. Some more facts on mucopolysaccharides 3D is that it's basically from a family of lysosomal storage diseases caused by deficiencies of enzymes required for the catabolism of glycosaminoglycans. And people with this disorder either do not produce the 11 enzymes required, or they do produce enzymes, but they malfunction and they don't work properly. And over time, the glycosaminoglycans collect in the cells, blood, brain, spinal cord, and connective tissues. It causes permanent or cellular damage that affects a person's appearance, physical abilities, organ / system functioning, and mental development. And it's basically from a larger group of lysosomal storage disease.
MPS 3 is divided into types A, B, C, and D, which are distinguished by their genetic causes. People with MPS III usually live into adolescence or early adulthood. About 25,000 babies born in the US have some sort of this disease. When both parents one copy of the defective gene, each pregnancy carries an 1 in 4 chance of inheriting the disease. Thus, beginning to understand this disease can help those who are vulnerable to inheriting it.
I'm gonna be saying Jaden's part because his mic isn't working, but some MPS 3D treatments. So to this day, there isn't really like a cure for this for MPS 3D, but there are treatments that help. One option is to inject a vaccine into the brain that helps break down the compounds that the disease causes in the brain. Another option is enzyme replacement therapy. Enzyme replacement therapies are available for MSP1, 2, 4A, 6, and 7, and other types that are being tested. Even though the treatments can't slow down the disease manifestation, the burden of having the disease will always be there. So onto the reflection.
Good job.
This is our reflection slide. I wrote, I think that this experience was really interesting. I didn't know a lot about neurological diseases, but I got to learn new facts about it. I also enjoyed listening to the professor speak.
I thought it was very interesting learning about uncommon neurological diseases and their causes. And it was also very interesting to know how they are cured and how researchers are trying to find treatments for it.
The experience about learning about MPS 3D was very interesting as I was able to learn new things about a not well-known disease. And I also think that more research should be done on this disease as it is very harmful to the patient.
I wrote that through today's presentation on MPS 3D, I managed to learn and gain more information about this rare neurological disease. I think that understanding such a rare, unique diseases is really interesting as it allows me to learn more about people who are affected by this. I'll also say Jaden's part because his mic isn't working. He wrote that today's presentation from Professor Tsui-Fen Chou was super interesting and helped me realize that the rare disease that has no treatment can be a big burden on humans that can ruin their lives. This shows that just one area in your body can make a huge difference. And that's it for our presentation.
Okay, thank you, Darren. Or thank you, Group 3. So for now on, I think starting from Group 4, could you guys just have one person to present the reflection side, so we don't like actually take that long to present each group. So so one of you guys can just volunteer and then share your reflection. Yeah,
right,
so
Nina
starting from
can
Group
do
4.
that.
Okay, yeah, thank you.
Hi,
Well,
everyone.
yeah.
My name is Nina Wu, and people in my group are Calvin, Richard, Lydia, Curtis, and Nancy. We are Group 4, LCCRNN.
Sorry, can you turn on slideshow format? Thank you. Great.
So the key points of today's speech, it's called starting off with a name that I don't know how to pronounce, so I'm just gonna skip it for now. It's just gonna call it MSD. MSD IIID is a rare neurological disease that makes the afflicted person unable to break down large sugar molecules called GAGs, because I don't know how to pronounce that name either. This is a result of a defect in the enzyme glucosamine 6-sulfatase, causing the lysosomes to be unable to process the GAGs. And as a result, this disrupts cell functions, which cause symptoms such as muscular or behavioral problems. There we go.
More facts about neurological disorders. There are many neurological disorders. Some disorders can affect our daily lives, from mood swings to hindering us from doing normal day activities like having problems with mobility. Most people also have trouble learning and remembering things and have issues with breathing and swallowing. Neurological disorders also cause chronic pain as well as shorten our life expectancies.
Neurological disorders are the #1 cause of disabilities in the world, and more than 1 billion people throughout the world are affected by some type of neurological disease. Nearly 7 million people die every year as a result of a neurological disorder, and roughly 10% of all emergency room visits are for some type of neurological complication.
The method of treatment is the enzyme or protein replacement therapy. It is a process that produces drugs to purify enzymes and protein.
Okay, so Lydia said, I learned that among the symptoms of neurological disorders, TTH, strokes, and more were prevalent. Calvin said, the presentation today taught me how the neurological disorder known as MSD-3D affected people and possible treatment options for these people. It may be interested in learning more about these types of disorders as he learned something new today. Curtis said, today I learned many neurological disorders cause millions of deaths each year, as well as put many people through extreme pain. These can include TTH, stroke, and BAT. I found the presentation today interesting as I learned more details about neurological disorders. I said millions of people die from neurological disorders every year, and it is the #1 leading cause of death. Because it is such an important issue to address, I found today's presentation very inspiring. Nancy said, something I learned today was that one of the neurological disorders known as BAT affected many people that led to their death. Richard said, today I learned that many neurological disorders are responsible for millions of deaths each year. Many of these disorders can hinder people from doing daily activities. Thank you for listening.
Great, thank you, Group 4. So, good Group 5. Start your presentation.
Are you guys able to see it?
I think you're sharing the, oh yeah, this one. You can click on the slideshow now. Yeah, okay, thank you.
Okay, so our group is called the 5 Good Bananas. We'll be presenting the CANDO neurological disorders and their challenges.
Some key points are that this disease is caused by the absence of an enzyme needed to break down a certain sugar molecule. It is an inherited disease and needs to be passed down. Some symptoms may include being unable to walk or the inability to function properly overall.
Some facts about MPS IID. Affected individuals could have deafness and loss of vision, progressive dementia, aggressive behavior, hyperactivity, seizures, inability to sleep, developmental regression, and intellectual decline.
So some individuals with MPS 3D have features that are similar to ASD or autism spectrum disorder, such as like anxiety, destructiveness, and aggressive behavior. As we further understand this, because it has similar symptoms to other existing diseases and can develop starting early childhood all the way until adulthood, we're able to figure out symptoms and hopefully viable treatments and sympathize with those who have MPS 3D.
Okay, and there's currently 2 possible treatment methods that are like in circulation. The one that was talked about today was enzyme replacement, also known as ERT, and the other one, which wasn't mentioned but is still like prevalent, is bone marrow transplant. And both these methods have been like studied and practiced excessively. However, there is still not a solid, like there's still no fully successful treatment method available.
Thank you.
Can just one of you group leaders present this reflection?
Okay. So for the reflection, I said today's speech was very informative and interesting. This is my second CANDO meeting, and it was our pleasure to have Professor Chou take her time to speak to us.
And for Melanie, this was my first CANDO meeting and I learned a lot from Professor Chou, and not only MPS 3D, but also in general about the process of identifying and examining diseases. And for Ethan, though today was my first CANDO meeting, it was very insightful and a great opportunity to learn about new things. I'm thankful to Professor Chou for taking her time to speak with us. And Wendy said, today's presentation was highly informative and it was great getting to learn more about this disease. And Henry, the presentation was very in-depth and professional. I was truly able to grasp the information on MPS 3D and learn about the effect it has.
Okay, thank you, Group 5. So could Group 6 continue their presentation?
Yeah, let me share my screen.
Okay. And same for like the reflection. If if like the group leaders would like to share their reflections, that'd be fun too.
Okay.
Good. There you go.
It's not okay. There we go. Who is okay? I'll do it. All right. MPS 3D. Group 6, and our team name is the White Mice. Our group members are Christopher Chen, Cindy Chang, Ted Hong, Ran Fu, and me.
Okay, so some major causes of MPS 3D is the deficiency of N-acetylglucosamine 6 sulfatase. And MPS 3D is basically a disorder which causes the body to be unable to break down larger sugar molecules called glucosamine aminoglycans, and more specifically heparan sulfate. And some of its major causes are progressive dementia, aggressive behavior, seizures, deafness, and a loss of vision, and impairment of movement, like the inability to walk.
Good.
More facts. So MPS 3D usually don't show any features until early childhood. MPS 3 is divided into 4 types: 3A, 3B, 3C, and 3D. MPS 3 is inheritable. Children with MPS 3 often experience sleep disturbances. It's important to understand this disease because it enhances our knowledge of science. It also gives us an insight into what the patients experience daily.
So there is currently no cure for MPS type 3D yet, but some treatments to improve the condition include enzyme replacement therapy and gene therapy. And these 2 treatments are still under development. So for enzyme replacement therapy, it involves the replacement of the missing enzyme with lab-based enzyme via injection. And these injections are administered into the bloodstream or directly into the brain via an implanted port. And this method of treatment is challenging and has a relatively high rate of failure due to the low amount of enzymes that actually successfully enter the brain. For gene therapy involves introducing a gene into the body which contains an instruction set to produce missing enzyme via injection of a virus. And this happens only once and is either injected directly into the brain, into the cerebrospinal fluid, or into the bloodstream. And this virus injected is mostly harmless and acts as a carrier of the gene. And this virus is usually AAV. And also, Professor Chou remarked that MPS type 3D is really hard to diagnose. And with the advancement of medicine, more patients will be found in the future. With an increase in patients, big pharma companies will jump onto the bandwagon and conduct research needed for hopefully better treatments or even cures.
All right, I will be reading mine and Sydney's reflections. And for today's presentations, I learned about a disorder I didn't even know existed. And I also learned how thankful I should be being able to walk, see, hear, and hear properly. And Sydney said, because of today's presentation, I was able to learn about MPS 3D as a whole. It causes symptoms, effects, and etc. If I had not attended this, I probably would not have been able to learn and been aware about this rare disease. Thank you.
Good.
Okay, thank you, Group 6. So will Group 7 do their last presentation?
Okay, let me share my screen.
Okay, thank you.
Wow. That's awesome.
Okay, so Chung, you can go ahead and start us off.
Okay, hello everyone. Our project is the CANDO project. We're Group 7, also known as, our group name is MPS Task Force, by Patrick, Amelia, Maya, Caitlin, Nadia, and Weber, and myself.
So the key points from today's presentation, we were introduced to MPS 3D, which is basically the absence of the enzyme GNS, which is a protein that helps the lysosome break down large complex substrates called GAGs. The result of this absence is the buildup of these substrates in the brain. And so these lead to the symptoms such as loss of bodily functions over time, such as walking and progressive dementia. But since this specific category of MPS is milder, the symptoms don't show up like super early on, so it's hard to detect. Finding a treatment, as Professor Chou explained to us, first they thought of gene therapy, but since that was not effective, they turned to enzyme replacement therapy. And the way they tested these solutions are on rats as models of human brains, but also she said that they were using pluripotent stem cells to create a mini brain, which I thought was really awesome, to test yeah.
Okay, next.
I think Caitlin or
Thank you.
Caitlin or Amelia.
Okay, so a little bit more about MPS 3D is that it's very hard to diagnose for a couple of reasons. Professor Cho mentioned earlier that some doctors, they don't really know what they're looking for because it's so rare. So that's one of the reasons. But there's also a lot of lack of information about the disease just because it is so rare. And also certain symptoms that point to the disease can also point to a lot of other diseases. So there's ambiguity in that. MPS 3D is an inherited autosomal recessive disorder. So you have to have the gene from both of your parents, which is part of the reason why it's so rare. And it usually appears before the age of 10. It's important for us to understand this disease because it's always good to be informed, but also it can affect people around us. Even if we're not aware of the disease, like maybe you're not diagnosed with it, but it's always good to be able to sympathize with those people.
So for the treatment introduced for the actual disease, the professor talked about how they use proteomics analysis to identify like biomarkers and use like biostatistics and informatics to like actually identify a treatment. So the treatment can be used like when a particular protein stops functioning, they can like replace the protein. And Weber.
And protein enzyme replacement therapy can help treat diseases. And it is a it is a costly process. Manufacturing drugs that can help influence proteins and enzymes, as well as ensuring the purity of proteins or enzymes are expensive.
And then Nadia can share her reflection.
Yeah, so I wrote way too much, but basically I thought that this was a very engaging talk, and my main takeaway is that there are always going to be new problems to solve. And being able to engage in that process of curing people can be really satisfying and awesome. And also just the vast applications of proteomics and protein treatment is just so fascinating. And it's just such an exciting field that you could be in. Like it could be cancer or neurological disease, and I think that's really cool.
And that's the end of our presentation.
Okay, thank you, Group 7, for your presentation. So before we end, So before we end, I'll be announcing our October CANDO event. So we'll be having Will also from Caltech. So he's also from the same lab as Professor Chou, and he'll be talking about Parkinson's disease, which is more common. And if you guys are interested, I'll be announcing the sign up information later. So, and then for like the upcoming months, I'm thinking of inviting like actual patients from patients associations, but I'm still working on that. So if any news come up, then I'll be announcing that too. So before we go, let's all take a group picture. So if you guys could just open up your camera, and then we all can take a group picture before we go. Okay.
So let me say, okay, Edmund say 1, 2, 3.
Okay, 3, 2, 1.
Cheese.
Cheese.
Hey, that's the first page, okay? Wait. And the second page, everyone, can you? Yeah, like thumbs up, okay? Okay, 3, 2, 1, cheese. Thank you. And how about the others that didn't come into the turn on their page? Okay.
Yes.
Yeah. Great. Again, that's thanks to Edmund to host this event, and how everyone think about it. Everyone like.
I know it's a bit long, but like thank you guys for staying till the end.
Yeah.
And
so
hope you guys. Oh, yeah.
About the attendance today.
Oh, attendance, yes. So about attendance, because we exceed 5 p.m., which is our like our original end time, but so you guys will be receiving an extra half hour for staying till the end. So yes, they'll be added to your attendance. And Sigu, should I do the event reminder, or is it okay? Because I don't know, I just included this from the weekly reminder, or like friendly reminder.
So that's it.
No. Sigu, I said, should I still talk about like the upcoming event reminder?
Sorry, yeah.
Okay, yes. Okay, so for the CSALs, like for Inland CSAL members, we'll be having our team bonding event tomorrow morning at the Tsujiwona campus. It's from 9:15 a.m. to 12 p.m. So we'll be playing some fun team like games, and then you'll learn how to teamwork, and it'll be a fun day. So, and for the rest, for TSTT, the mentor applications due at September 14th. So like if you want to be a TSTT mentor, then please apply using the Google form that's in the weekly friendly reminder. And then the Inland CSAL Music Ensemble will be having a rehearsal tomorrow at 1 p.m. And for close for costs, and then those who sign up, then you'll be washing clothes, and then you'll return to Jason on the medical outreach site. So September 25th. Yeah. So that's it for today. It's like an entire CANDO event. So thank you guys all for being here today and staying till the end. And
yeah, remember tomorrow morning